My name is Craig Thomas. Good morning, or afternoon, or evening, depending on when you're watching this. It's my pleasure to welcome you to the NIH's Principles in Clinical Pharmacology course. I've been asked to give the introduction to Module 1, and as by extension, the entire course. Before we begin with this introduction, I think it's worthy to say that you're all in a very fortunate position to be able to watch and learn from each individual lecture in each of the modules. You're going to get a wealth of information about the type of science that involves pharmacology and clinical pharmacology, about the kind of data that is generated, about how that data is used collectively to administer drugs to yield an optimal clinical benefit to patients. As this is the introduction to Module 1 in the course, I think it's imperative that we begin with some fairly straightforward definitions of what pharmacology is. Pharmacology is the science of how drugs act on biological systems and how that system responds to the drug. A system is a fairly broad term. It can mean anything as complex as the human body, which is a complex and integrated system. But it doesn't have to be such a complex system to render any kind of pharmacological useful data. It can be something as simple as a cell line or a protein that could be used to generate data that can be used by pharmacologists, clinical pharmacologists, and the practitioners of medicine to better optimize how a therapy might be used. It's important to realize that within each of the lectures, within each of the modules, you're going to be learning about the kind of data that is generated in these various systems. You're going to be learning about the details of how that data is generated, how it's used to better define therapeutic use, alone or in combination with other drugs. Clinical pharmacology is the applied version of that. It's how all of that data can then be used by physicians, by veterinarians, by pharmacists, by scientists to better practice medicine. So both the basic research within pharmacology and the more applied research within clinical pharmacology serves as the basis for the lectures you're going to hear throughout this course. And basically this bullet point gets to that point. So doctors, veterinarians, pharmacists use this knowledge to achieve optimal therapeutic outcomes through the appropriate preparation and dispensing of medicines. And that's important. So it's not just how they're given, but it's also how the drugs are prepared before they're given. So this is complex, and throughout the course you're going to see the complexities laid out by the lecturers. It's important, at least for me, and I think for a lot of people whom taught me and whom I respect in this field, to bring this down to a core principle. At the heart of this is mechanism. Modern drug discovery is a mechanism-driven field. And it's important to realize that every therapeutic, nearly all new experimental therapeutics, and I would probably say all, although I just can't prove that, so I'll qualify it with nearly all new experimental therapeutics, have a defined mechanism, sometimes more than one mechanism. And most have a companion diagnostic that allow researchers to track the mechanistic engagement of that medicine within the complex systems like the human body. And because we have a mechanistic rationale or belief that we have a mechanistic rationale for why a medicine should work within a human body to alleviate the effects of a specific disease, and because we can oftentimes track whether or not that mechanism is engaged when a therapeutic is given to a human being, because of these reasons, because of these realities, clinical pharmacology has never been more important in our ability to understand the benefit of a medicine and to tweak it, to utilize it more effectively. So really, I want to restate that the lessons you're going to be learning throughout all of these modules are imperative to anybody who wants to practice clinical pharmacology. This gets to the concept of seeing the forest, seeing the trees, and this is not an easy thing to accomplish in any complex field, economics, international relations, and certainly pharmacology and clinical pharmacology. There is that battle between seeing the forest and also seeing the trees. Now, what do we mean by the forest, and what do we mean by the trees here in pharmacology and clinical pharmacology? So the forest is the concept of visualizing, understanding any medicine with a clinical benefit. So a large percentage of the population takes statins to control their cholesterol, certain types of cholesterol, circulating cholesterol in the blood. And physicians can see that effect take place in an individual patient. And that's the forest. That's seeing the benefit of a medicine. In the sense of a population, we can see populations of individuals taking statins. We can see the effect of how cholesterol, certain types of cholesterol being lowered in a population. That's the forest. But why do statins work? Why does that specific medicine work? Why does it effectively bring down cholesterol levels in an individual, in a human being? That's the trees. The reason behind why these medicines work, that's the more detailed oriented version of seeing the trees. Now, any scientist wishing to understand, practice, and master clinical pharmacology must be able to see both the forest and the trees. And it's important within the lectures of Module 1 and all the others. These are designed to help students learn about both. Now, within each lecture, you're going to be learning specifics. You're going to be learning about the trees. Now, it's important to keep in mind the forest. There are going to be individuals who are in medical school watching this course. You're going to be people who are eventually going to be visualizing the forest. It's imperative that you also understand the basics, the trees, the real details, the kind of data that pharmacology and clinical pharmacology yields. And that's the kind of data that you're going to be learning about in each one of these lectures. So let's consider some of the core principles. Foremost, identifying and developing an experimental drug with a defined mechanism of action is oftentimes the first step in developing a new therapy. This is drug discovery. This is where we're going to be starting with a lot of these lectures. As a matter of fact, all of Module 6 delves into drug discovery and development. Within that module, you're going to be learning a lot of how scientists and pharmaceutical companies, academic labs, begin the process of developing a new drug. Almost exclusively, that process begins with defining a specific mechanism of action. So these scientists, and these are high-level practitioners of this type of science that do this and do this well, they're going to be looking for a drug, a small molecule, a biologic, which inhibits an enzyme or activates a receptor. That's the defined mechanism of action. And the theories behind why inhibiting that enzyme or activating that receptor might alleviate the symptoms or causes of a disease is the common first step that most drug discovery efforts take place. And that's when data that would broadly be referred to as pharmacological data, that's when that data starts to get generated. And you're going to hear about a lot of that early data in the Module 6 drug discovery lectures. Within that mechanism of action, that's when we start to track a lot of this types of data. And we try to anticipate, before we even get started, what are the potential consequences of that mechanism of action, of inhibiting that enzyme in a disease cell, in tissues, in the system at large. Because it's within that anticipated consequences of inhibiting that enzyme or activating that receptor that lay the groundwork for the potential efficacy of a future drug. Now, we also try to anticipate the consequences of inhibiting that enzyme or activating that receptor, anticipate the consequences of the mechanism of action of that drug, or future drug, on healthy cells, on healthy tissues, on the system at large. Because it's oftentimes that mechanism of action working systemically that creates the dose-limiting toxicities that will define how much of a drug can be given to an individual patient, the potential toxicology. So beyond that, we have a handful of mechanism of actions that we know can't be tolerated by healthy cells, by the tissues, by the systems. These are mechanism of actions that we've learned through trial and error, hard-fought lessons, lessons that doomed many drugs. If we have an off-target event at one of those mechanisms, we know that molecule or that potential drug is probably not going to survive the development phase of a drug. So these are known toxicities. Common examples include the hERG channel. This is an ion channel that governs certain elements of the human heartbeat. And we know that we can't inhibit or modulate that particular ion channel in any relevant way and still have a therapy that would be of use with zero toxicology or zero toxicity. So there are hard-fought lessons on known mechanisms of actions. These are dozens, if not almost 100 known mechanisms of actions that we know that a future medicine can't have as an off-target effect. So we have all of these concepts, the anticipated consequences, good positive consequences of the mechanism of action that we're interested in, which would yield a potential efficacy in a patient, the potential that that mechanism of action might also have toxicity, and then known toxicities. All of these are going to be the kind of data that surround the question of mechanism of action for a future therapy, and the kind of data that pharmacologists and clinical pharmacologists rely upon to then utilize a future medicine in a disease population. And it's important to state that all of these mechanism of action related outcomes are concentration dependent. So we might need a concentration level to yield that efficacy, but we can't achieve a certain concentration level of that drug in that human before it creates a toxicity. Within that dose window that achieves the desired mechanism of action related effect, but not the toxicity, that's the critical element that defines the therapeutic index of that therapy. The scale and timing of that mechanism of action related event, so the drug's effect in the body, its anticipated mechanistically defined effect in the body, this is critical. So when you provide a drug to a human body, it's not there forever, and it's not there at a constant concentration. You're going to see lectures on pharmacokinetics that define how the different pharmacokinetic principles of a drug define how long it's there, what kind of concentration it achieves. And not only what kind of a concentration it achieves in the blood, where we're able to track it, but whether or not that drug systemically distributes to all the tissues of the body. Does it go to the brain? These are all things that we can design within our drugs. These are all things that we can track within different systems, animal systems, or even in the human body. And it's important to understand that the scale of that mechanism of action, the level of inhibiting it, plays a real role. So what percent of that mechanism of action in the disease cells, in the disease tissue, in the system at large, is needed to achieve an effect, and a positive effect for that patient population? Is 50% enough? Do we need to achieve 90% inhibition of that enzyme or activation of that receptor to achieve a benefit for that medicine? These are things that need to be thought about, that pharmacological data and clinical pharmacology data will provide evidence of, because that's going to affect how eventually we dose that drug into patients. Not only that, we need to know about the level of effect on these off-target actions that define whether or not this drug is going to be safe for the human being. So again, 50% inhibition. Could we tolerate a certain amount of inhibition of the hERG channel? Perhaps, but these are kind of the questions that the teams that are developing a drug need to ask themselves. How long can the mechanism of action or the off-target actions be tolerated in the system, and how long does it need to be in place for it to be efficacious? Drugs, again, small molecules in particular, rarely are there for days or weeks. Some biologics can be, but it's important that we recognize or understand that the duration for the drug in the human body and the duration for which it's affecting that mechanism of action, the inhibition of that enzyme, plays a real role in how we're going to define how useful the drug is, and when it's dosed, the schedule that it's dosed upon. That's all things that need to be understood and are part of the core principles of pharmacology and clinical pharmacology. So it's important that you understand as you listen to the lectures and all of the modules that this is not just a binary on-off system. The presence and actions of a drug and its mechanism are transient. They're defined by the exposure of that drug, the pharmacokinetically defined exposure of that drug, and the activity of that drug, the pharmacodynamics. And you're going to learn a lot about pharmacokinetics in modules 2 and 3. You're going to learn a lot about pharmacodynamics in modules 5, and the lectures in module 5. So continue on with the core principles. You're going to be learning a lot about these particular bits of data. It's always important for you to keep the human element in the back of your mind. Different patient populations will have unique needs. We treat pediatric patient populations different than we treat adult populations, different than we treat geriatric populations. They will all have different abilities to deal with certain levels of toxicity of a drug. They're going to have different metabolisms that define how quickly a drug is absorbed or eliminated from the body. All of these things are things that keep in the back of your mind as you listen to these individual modules, but furthermore, as you become a practicing scientist within this space, don't begin one of these programs without asking yourself, what is the anticipated population? If it's a geriatric population, you have to consider the metabolic and patient-derived demands of that population. It's going to have a lot of effects on the ideal type of drug that you're going to be developing. Different indications will have unique tolerances. For instance, cancer therapies are broadly considered to allow a lot more toxicity than therapies that we would use to chronically treat anxiety. Individuals who are up against a very difficult cancer diagnosis might be more willing, and typically are more willing, to deal with therapies which cause a higher level of adverse effects. Whereas somebody dealing with a mild level of anxiety simply doesn't want to be dealing with a very toxic therapeutic regimen. The growing field of pharmacogenomics. This is basically how a patient's genome is going to influence the choice of therapy and the outcome of a particular therapy. This is a rapidly developing field. You're lucky that there are, in fact, a specific set of lectures within modules 4 and within modules 7 that deal with these particular aspects in this burgeoning scientific field. It's remarkable that we have as much knowledge as we do today about a human being's individual genome, and we're learning more about what that means for how an individual's genomic disposition affects how specific therapies may or may not work. Ultimately, the goal of all of this, ultimately the goal of teams of scientists, whether they're medicinal chemists or pharmacologists or clinical pharmacologists or practitioners of medicine, the goal is to use this type of data to help doctors treat the patient rather than disease. We're learning more and more that individual therapies are probably going to be the future of medicine, and a lot of the lessons you're going to be learning within each of the modules, each of the lectures, is designed to help you understand how a specific medicine, how the data that is broadly characterized as pharmacological data or clinical pharmacological data can then be used to define a therapy course for the person, the patient, the individual, not broadly the disease. So clinical pharmacology will play a critical role in making this a reality. As I stated at the beginning of the course, this is complex, and I made a point to say very, very complex, and it is. I don't want that to be a daunting element. It shouldn't be a daunting element to anybody listening to these lectures. This complexity is not insurmountable. Individuals do it all the time. They learn about these individual data sets, the types of data. They become expert practitioners in pharmacokinetics or pharmacodynamics. They become experts in medicinal chemistry or molecular biology, cellular biology. They become expert practitioners of experimental medicine. Because even though this is complex, it's not insurmountably complex. But it's important that we embrace that complexity. Complexity is not a vice. Complexity is defined by the fact that humans are complex. Understanding the principles of clinical pharmacology allows you to appreciate that the reasons behind success and failure can be narrow. Perhaps the drug was not dosed at the right schedule or dose. So it can be a razor-thin margin of error that defines success or failure for an individual therapy. Oftentimes this is not clear. But as we develop the core principles of pharmacology and clinical pharmacology, we're getting a better grasp of what's going to define success or failure for an individual therapy. Drug discovery, pharmacology, clinical pharmacology, and clinical practice, as a result, are team sports. No one individual, or if one individual can learn all of this and be an expert practitioner of all of it, that's pretty rare. As a result, it's usually teams that are able to accomplish this. Oftentimes that's within private industry, at biotechs and pharmas. A lot of government and really strong academic centers can do this as well. But oftentimes, or not oftentimes, almost always, I'm going to go so far as to say always, this is a discipline that is defined by teams of scientists coming together to develop a therapy. And it's allowing the individuals who are experts in one element of it to be driving the project when it's at that stage, allowing solid professionals to do that work. That's where success comes from, when teams of experts come together with a common purpose. And it's important to say that success or failure is oftentimes defined by the application of best practices in all domains, all the way from target validation through the clinical application and advanced phase 3 clinical trials. Best practices and having individuals who are very experienced and knowledgeable in each element of this process, really that is going to define whether a therapy's development achieves success. And as I stated, although this is complex, it's not insurmountable. Last year, in 2018, the FDA approved 59 drugs, giving hope to patients with complicated diseases ranging from cystic fibrosis to migraines. Each of these therapies is a testament to the hard work of professionals who, like you, started off by learning the principles laid out in this course. So really, you're at a terrific position. You're at the beginning of an exciting adventure, learning all of these lessons that are laid out in the courses, modules, and lectures coming forward. That's the extent of my introduction to these lectures. I hope that I know you're going to find them all to be stimulating. They're all going to be of broad interest to all of you. Some of them are going to be of specific interest to several of you who are interested in developing and learning about pharmacokinetics, pharmacodynamics, drug discovery in clinical practice. Embrace these lectures. Watch them, rewatch them, and allow them to stimulate your interest more so than it already is. Thanks for your time. Enjoy all that's to come.